Okay, thank you everybody for being here today, and it's a very nice platform for us to have because we're able to talk to a global audience, and we would like to tell you a little bit about what is Euro-BioImaging about and how we are offering image analysis and image data related services, which could be of interest to you all. So starting up, let's start with the, okay, I should have checked that. Yeah. So let's start a bit with the introduction. So I am Aastha Mathur. I'm the coordinator of image data services at Euro-BioImaging. And today I've been joined by two of my fantastic colleagues. We have Stefan Klein, who's an associate professor at the Radiology and Nuclear Medicine Department at the Erasmus Medical Center. And he's also part of our population imaging node at Rotterdam. And we also have Jan Sporing, who is a professor at the Department of Computer Science at the University of Copenhagen, and a part of our Danish bioimaging node. Actually, this is not all. I think some of you might have already heard from a couple of people who are associated with Euro-BioImaging. For example, we had already a talk from Perrine Pogiloto and Jean-Yves Teneves yesterday, who are part of the France Bioimaging, and we had Anna and probably Carolina Walby from the Swedish National Microscope Initiative, who also gave talks about their cool tools that they're developing for image analysis. So it was nice to have a lot of nodes represented in this meeting. So that was pretty cool. And how do we manage all this? Actually, we manage all this because we are very big. So Euro-BioImaging is actually a European research infrastructure for biological and biomedical imaging and analysis technologies. It is distributed in nature. We have 149 sites across Europe, and these sites are imaging facilities that are parts of national universities, national research center or international research centers. And with all this power of the distributed structure of Euro-BioImaging, we're able to offer more than 50 technologies to our users. Now, this big bunch of people also require some coordination activities, and our coordination activities are taken care of by what's called a hub. Our hub is also distributed in nature with our seat in Turku in Finland, the biology hub at EMBL in Heidelberg in Germany, and our med hub, which is in Torino in Italy. All right, but what do all these people come together and offer you as a researcher? Well, for a researcher, Euro-BioImaging offers open access to biological and biomedical imaging technologies. And very importantly, along with that comes support from our expert technical staff at these facilities. Because with the imaging technologies getting so advanced, this support is essential, if not very crucial in most of the cases. Along with the access to instrumentation, we also provide image data services for analysis, storage, and sharing. And I will come back to that a little bit more in details. Apart from that, we are also offering training opportunities for individuals, individual users and researchers, as well as facility staff. So I want to discuss you a little bit more in details about the image data services. And in general, it can be, let's say, split into two parts. Image data services at Euro-BioImaging can be offered at the nodes, which is basically a user coming up with a project. Maybe they have their own data and they can contact any of the experts in bioimage analysis at many of our facilities, two of whom are actually here with me today, and they will talk to us more about how they offer services at the Euro-BioImaging nodes. But apart from the data analysis service at nodes, we also manage a few things at the hub. So at the hub, we provide support to the nodes in order for them to be able to provide these services directly to the users. And in terms of the services that we offer, we do not directly work with individual user projects, but actually develop methods and technical support for services that would be more useful for the community as a whole. So let's get a little bit more into what we do at the Hub. So starting with all the things that we are involved in, one of those is representing interest of imaging technologies and image data at the European landscape. And we do this by being a member of the European Open Science Cloud Association and helping in its governance through the task forces. We are also associated in a lot of the EOSC projects, or European Open Science Cloud. It just gets very long over time. So EOSC projects like EOSC Live, EOSC Future, BioCovid, Healthy Cloud, etc. So in addition to that, we are also supporting open image data repositories like the BioImage Archive, and maybe some of you had a chance to hear the talk from Matthew Hartley earlier in this conference, as well as the IDR and Empire for electron microscopy data. We also coordinate with community initiatives like Corep-Limi, which is focusing on quality control, and especially we are involved in the metadata working group. And additionally, we support image data service provision by hosting expert groups where our experts can meet and discuss challenges in provision of image data analysis as a service, as well as some technical support. Now, what I've done here is that there are a lot of things here. So I've tried to pick out a couple of examples to demonstrate the kind of efforts we've been involved in. So first thing that I picked was the work that Jean-Karim Harish and Beatriz Serrano Solano at our EMBL node did as a part of an EOSC Life project, which was to basically provide cloud tools for reusing open image data. So they used data sets that were coming from screening like large genetic screens, but they use it for something different. They used it for looking at nucleoli and their distribution in the cells and the morphology, etc. What they ended up doing in practice was to make this fantastic pipeline for data analysis, which comprised of different modules, where first they had to take care of data access, and this was done through the Human Protein Atlas and the IDR. Then they had a module for segmentation and feature extraction, which made use of Cell Profiler. This was all combined in an automated workflow that was made possible by using Galaxy, and it also had a component of a downstream biological analysis, which was basically a Jupyter notebook. Now, getting a little bit deeper into the automated workflow, you'll see that in Galaxy, all this structure is very modular. So you can have like a module for import, and you can have something for nuclei segmentation, for the nucleoli, for background, feature extraction, etc. And all this was then packaged in this workflow that could actually be run using cloud resources. Well, with this, what they demonstrated was scalable mining of the image repositories. And in addition to just be able to get the data out of these repositories through Galaxy, they also brought or ported a lot of image analysis tools which were belonging to the Cell Profiler suite into Galaxy so that people can use them now. In addition, they also brought basically image data to Galaxy community, because Galaxy is often and very widely used by the omics community for transcriptomics and genomics data analysis. So this was one of the projects that has already taken place. And if you want to have a look at the tutorial, look at the QR code here. It's very nicely explained if you want to have a look at it. But now I also wanted to briefly mention where we are going. So here I picked one of our ongoing projects, which is part of the EOSC Future project. And here the lead is taken by Bura Ozdemir, who is the image data specialist at the Euro-BioImaging Hub. Now he is trying to implement this idealistic workflow for image data that we can imagine, that there is large and information-rich data that needs to be put in a standardized file and metadata format. Once it's all standardized, it should be compatible with some cloud workflows for doing analysis, and then it should be available openly in a fair image data resource. What we are doing here is we're working together with the community and a lot of things that have already been developed. So we are extending the OME.SAR format to include high content microscopy data in a format where a lot of metadata can also be stored. In addition, we're going to make use of the things that we did in the EOS Live project, like I talked about before, and use the modules from Self-Profiler to do some image analysis, and then eventually push this data to the bioimage archive. This process would be, again, implemented in Galaxy as a Galaxy pipeline. And here we're really trying to enable that the whole community benefits from each and every step in this workflow. Well, I just wanted to summarize by saying that what we are trying to do at the Hub and together with our nodes is really to have image data from our users move to this fair and open image data space. And once it's there, it can be used via data analysis pipelines and probably even new method development so that you can extract new knowledge and new methods from this very powerful data. In addition to that, a very important part is here where the users themselves are helped by our experts at the nodes to generate new knowledge from the images, well, I2K, I guess. And for that, we have two of our colleagues, and I think it's Stefan Klein, who's going to tell you about the things that they're doing with the medical image data service provision. So I'm going to stop sharing. In the meantime, if you guys have any direct questions, please go ahead and ask. I will try to answer them. And if that does not seem to be the case, we can probably just wait for the discussion at the end. And Stefan, please take it away.
Okay, thank you very much for a nice introduction also in your slides. And for the overview of Euro-BioImaging. So my name is Stefan Klein. I'm an associate professor in medical image analysis and machine learning at the Department of Radiology and Nuclear Medicine at the Erasmus Medical Center in Rotterdam, the Netherlands. And I would like to give you a bit of an overview about the services we offer from the European Population Imaging Infrastructure. So that's the population imaging node of Euro-BioImaging. For that, I will start with a very quick background on the type of research that I'm doing in my own with my own team, and there's a large community of people doing this kind of research, of course. And then I would like to show some examples of the infrastructure that we developed in order to streamline and consolidate this type of research to facilitate it. And finally, I like to point out how what we're trying to do to make this infrastructure available for others, because we put a lot of work in it, and we hope that others can benefit from it, and maybe you even. So the research that we're doing. So as I said, I'm a researcher at the Department of Radiology. So mostly I'm focusing on MRI scans and CT scans of humans. Actually, mostly it's mostly in the clinical domain, but the techniques that we are working on also apply to preclinical images. So we start with images, and the main aim of our research is to develop tools to extract more information out of these images. So for example, segmentation or to combine images using image registration. So to combine multimodal images or we develop models from large numbers of large data sets of images from multiple patients in order to create models of disease. For example, you see here an example of Alzheimer's disease, where you can see how the brain is changing as a function of due to the disease actually. And more we are actually trying to take the image as an input and then train a model that predicts a kind of biological characteristics that underlies the or causes the appearance on the image. For example, when we get an image of a tumor, we try to predict what are the genetic mutations of the tumor, or what is the tumor phenotype, or is it a benign or a malignant tumor, or we can even try to predict what is the prognosis of the patient based on how the disease looks. For example, how severe is the brain atrophy in case of dementia. And obviously for this we use a lot of machine learning technologies, but also more classical image processing techniques for segmentation and registration. I'm showing here examples really from the radiology domain, but we also developing similar techniques in obstetrics and in gynecology, where we have ultrasound images, 3D ultrasound images, and we try to combine them and first again do a segmentation of the embryo in these kind of images, and also we try to build models of how how babies are growing during the pregnancy using automated image analysis. And a third example where imaging also plays a role is ophthalmology. So in imaging of the eye in ophthalmology departments, a lot of imaging is performed because the eye is a perfect way to look inside. It's made for looking inside almost. So here you see a few examples of optical imaging, where also again we can apply segmentation, registration, motion compensation, all these kind of techniques. So it is quite broad, and you can imagine that you could even extend to pathology imaging, for example. So in this kind of research, as I said, we do a lot of machine learning. So what is important to organize your data, because if you talk about big data, you need to first make sure the data is accessible. So, and that gives us, we realize that gives us some infrastructural challenges, because first you need to collect the data, and often that's in our field we collect data from multiple institutes to have as much data as possible as representative. Of course, this data needs to be anonymized because it's data of patients. So we need to, that's the very first thing that has to be done at the source of the data. Then typically we find in this clinical domain that data that we get is a mess. Unfortunately, it has to be cleaned up and structured, because it's not ideal academic research data with homogeneous protocols, but it's quite heterogeneous. So we need tools for that. We need to store the data, of course, in a place where close to the compute infrastructure, and also in a way where all the people who want to have access to the data can get access. So we need to have tools to share the data with others in a controlled and secure way, because it is patient data. So we need to be a bit careful often. Then we need tools to do inspection, so quality assurance and scoring of images by radiologists, and also to make manual annotations, because we need them for training these machine learning models. So this, we try to also to streamline these processes. And then, then actually the interesting part starts, the processing and the automated processing and the analysis. And of course, we also there's lots of room for streamlining that part of the research. And imaging data is never in research considered on itself. It's always in combination with other data, like clinical data, sometimes with genetics data. And so there need to be tools to integrate it in a traceable way with other data. And all these aspects, in order to ensure that our research is reproducible, we'd like to have a proper governance, so that to make sure that everything is traceable, and we can always go back to see if, where possible biases were introduced. And I put a number of logos on the bottom of this slide, because there are several projects, both national and European projects, and in which we have developed infrastructure to help with these steps. And that has led to a kind of overview picture that we typically use to organize it a bit. This is shows a bit the typical flow. We start with data on the left that comes from scanners or from the clinical database systems. Then we need the tool to anonymize. We use the clinical trial processor. Then we store the data. I will tell more about that aspect in a system. It's called XNAT. And then based on this storage, we connect multiple systems to it. For example, for viewing, for quality assurance, for annotation, and for automated processing. So this is a bit the overview picture. I will not go in detail on all these tools, but I would like to say a few more about the core of it, which is XNAT, which is where we store our data. And I think that's whenever you think about streamlining the research, it starts with the data. So we chose to adopt an existing system for that. As an open source package called XNAT, the Extensible Neuro Image Archiving Toolkit, out of the top of my heart. So that's a platform for storage, sharing, and management of medical image data. So it's open source, developed in the US, but there's a very active community around it. And many groups in our field are using it. So it's a kind of de facto standard, you could say. And the simple thing it does, it imposes a kind of structure, in the standard structure on how you store your data. So there's an organization, like there's projects. In each project, there's a list of subjects, so the patients or the volunteers. And then for each subject, there's several experiments, so those are the visits of the patient where they got scanned, or so you have an MRI scan and a CT scan, or at baseline and a follow-up. And then within each experiment, there are all the scans that belong to that experiment. It sounds straightforward, but by making it standard, it harmonizes a bit the data storage across projects. There's because we talk about sensitive data, so there's nice options for access control. So you can mark your project as a public, restricted, or private project, and there's different roles for the users. So giving them read and write roles. It supports both DICOM and non-DICOM data. So DICOM is the main standard in our field, and interestingly, so there's a visual interface, a GUI, but also a programming API. So we can, so it's useful both for the clinicians who don't like to program, but also for us informaticians who like to automate everything. So actually, we developed also a Python toolkit that wraps this API. So to make it really easy with a few lines of code to talk to this XNAT and to upload and download data, to organize the project, etc. So this software is really at the core of all of our research, where we to organize and manage all the data. The next component that I would like to highlight is the processing, the automated processing. And for that, we developed also a Python library called Faster. And Faster is an automated workflow engine. For an automated workflow engine for streamlining large-scale automated image processing. So it's also open source, and it's based on Python. But basically, we define networks of workflows, and these networks consist of nodes or modules that can actually wrap any executable code. So any executable can be used in it. So not only Python native code. And we are actually currently working on also supporting Docker containers in a seamless way, which is, yeah, I think the future for easy deployment everywhere. The nice thing is of Aastha that it's a kind of a modular platform, and there's also different execution engines. So when you can run it a processing pipeline on your own computer, so then it just works single-threaded. Or you can also run it on a large compute cluster, and then it will automatically schedule different jobs that can be run in parallel. And and you don't have to change anything in your code for that. On top of that, it has built-in data provenance, so it really produces a whole trace of which data was used in which processing flow, and it connects directly to the XNOT system that I showed on the previous slide. So we can really streamline the whole analysis and make it very reproducible in this way. I show you an example of some code on the bottom of for running a network. It's just for illustration; you don't have to go in detail. And an example of a very simple workflow, a simple network, in which we do a simple registration between two images using an existing open source package for image registration called Elastics. Using this faster, we developed a number of standardized pipelines that was basically based on our own research. So this was work based on PhD students in our team. But then we made some effort to consolidate that research, because we all know the problem that when a PhD student leaves, we don't have access to the code anymore. So now we asked them, okay, put your code into this as a module in this faster system. And because we did that effort, now after these students have left, we can still run those tools and offer them as a service actually to others, and also just for ourselves even. For if we get new data, we can run those pipelines again that provide tissue segmentation of brain structures, do some quantitative analysis of perfusion MRI, and here's an example of segmentation of knee MRI. So a number of these, we are developing a portfolio of standardized biomarkers, you could say. Also, more recently, we have also, on top of this faster framework, developed a software called WORC. It's that facilitates reproducible radiomics through automated machine learning, and that is we also made it open source, and this software completely automates entire experiments with machine learning on medical images. So all you have to do is basically point to the set of data and which ground truth labels or something that you want to predict or that you want to classify. And then the software will automatically create actually the whole machine learning pipeline, which is again a combination of different tools. We showed actually that on 12 different applications, so all kinds of different tumors, different imaging modalities, that with the same settings, it could produce very competitive machine learning models. So if you're interested, I think this actually has broader application with a few changes than just in radiology alone. And this is built on top of the faster software, so it also inherits the advantages of that package. Yes, so this was a summary of the infrastructure that we developed. So how are we offering this? So for this, we are benefiting from this whole this organizational structure of Euro-BioImaging, where we are hosting one of the nodes, the European Population Imaging Infrastructure, and so as a summary, this EPI2, it offers tools, services, and expertise for storage management and automated processing of medical images. And specifically, the technologies offered are what I just showed. So first of all, we offer this XNOT imaging archive and experience in using it. The XNOT Pi library, that's a Python library to interface with XNOT. The faster workflow engine, and then a number of validated pipelines for selected imaging biomarkers, and also for development of AI models, like I just showed. So how do we make this available? So how do we make this accessible? So we have 3 access modes. First is very simple, what I already showed. We just make the software open source. I mean, it's as easy as it sounds. And we try to write good documentation so that it's immediately useful for anyone. And we typically make the software available under Apache 2 license, so it can also even be used by companies. So I think, yeah, this it sounds trivial, but I think this is often the most impactful way of making things available. Second, so I already said XNOT is actually based on an existing system, so it is already open source. You can already download it, but if you want to install it yourself, I mean, this needs a lot of work if you want to host it and manage it. So we actually offer it also as a service that you can just use an existing XNOT and put your data there. So we are hosting one, and I will show that here in the next slide. So that's called at xnot.bmia.nl. So this is our main installation of XNOT. It's hosted by a professional company in the Netherlands, and that does regular backups and patches of the server. So it's a secure setup. There's also a full-time help desk for first-line support, so people can just go there and ask for to create a new project, to create new users, etc. So that I think there's quite a professional service around it. And to the access model it's based on, I mean, you have to pay a fee per year based on the size of your project to use it, and I think it's quite a fair price. It just covers the costs of actually the hosting of that company and contributes a bit to the personnel at the help desk. And mostly we find if people notice beforehand, they can include the budget for this in their applications for grants, for example, and then it's quite affordable. So the third way of giving access is actually to offer our image analysis expertise and pipeline and the biomarkers as a service. This is a bit more in development, I would say. But we try to formalize this more and more. So we have defined a number of phases where interested researchers from anywhere can approach us, and then we will first start with the intake message, so intake meeting where we discuss what the customer wants, and then we also make a budget, or we find we have to allocate resources for it. And then there are some obvious phases, like we do planning, then we make a decision, okay, can we offer this or not, and then the actual the processing starts, where we would first we have to get access to the data, we have to do the cleaning if it's necessary, then do the processing, do a quality check, and perhaps do the processing again until we are finalized. So via this image analysis as a service, we like to offer these consolidated biomarkers that we developed, and the use of this automated machine learning toolbox. We'd like to offer that as a service to any researchers who are interested in it. All right, I think this ends my talk. I hope I give you an overview of how we at the European Population Imaging Infrastructure make available our infrastructure for medical image analysis. I realize it is more, the examples are more from the radiology domain. So, and this audience might be more interested in microscopy, but I hope you find some links, analogies, and I hope it convinces you that some of these techniques might actually be applicable in your field. Thank you, and regards from our team in Rotterdam. Any questions?
Thank you so much, Stefan. That was fantastic. And there's a lot of tools. And indeed, I feel that when it comes to analysis, it's something that tends to span different fields. So definitely people will find interesting tools within your portfolio as well. So thank you very much.
Thank you.
Do we have any questions directly to Stefan's talk at this moment? Now, if that does not seem to be the case, maybe we can go to Jon's talk. Yeah, take it away.
Thank you very much for a nice talk, Stefan. So I will try to continue where you left off. So my name is Jon Sporing, and I'm a professor in computer science. And these last, well, 10, 15 years, I've been working a lot with image processing, where the images somehow involves microscopes. The last 4 years, I've had the opportunity to be part of this Center for Quantifying Images from MAXIV. And there we've had we've been able to offer data science, or I like to call it image science as a service. And this fits very well with what Aastha and Stefan has been talking about, how we can develop that as a service on a larger scale. For example, in Euro-BioImaging on a European scale. So what we are in the process of in Euro-BioImaging is to try to understand how we most successfully can offer image science as a service, as a node in Euro-BioImaging. Before I start, I just want to say a little bit about myself. So I think lately, whenever somebody has been telling me what I do, they say I'm an applied mathematician. I think I'm a computer scientist, but well, maybe I should revise that. But what I do a lot is I do method development, and this is a recent example. So what does that mean? Well, that means that we very much consider the complete pipeline. So from up here, this is an example of an electron microscope image of a rodent's brain. This is a public domain data set from Lausanne. There is often a segmentation step, and these days deep learning is the tool of choice. But even though deep learning does many good things for us, it still is a huge job for us as image scientists to do the ensuring analysis. And for this, so what you see here in blue, green, and red is a segmentation of the image to the left in 3D. The blue are the vesicles, so these small nodules here. The red are the active zones by the synapses. And then we were also, for this project, interested in mitochondria, which are in green. And then one of the questions that the collaborators were very much interested in was trying to quantify if there is any buildup of vesicles near the active zone, and in general, what are the geometric properties of structures inside cells. Since many people here are probably working with microscopy, I think it'll be clear to most that cellular structures vary enormously in shape, so we have to come up with something that is somehow agnostic to shape. And we came up with a method that is based on point processes. So if you think of these red squares here as stylistic examples of the active zone, and these round ones as vesicles, well, imagine that you, with mathematical morphology dilation, grow the red areas and then investigate well, it turns out 4 different intersections that you can have, and use these. So these intersections we've derived, and then you can come up with relations equations. So this is very much what I do. But these last 4 years, I've been part in this QIM. And this is one of the places where we're looking for inspiration on how to shape image science as a service. And I think, in contrast to what Stefan just was saying, I think I will focus a little bit more on people. But let me first tell you what QIM is. So MAXIV is a synchrotron. There's a small image up here. So it's a synchrotron facility which is in Lund. Lund is a city just 1 hour drive from Copenhagen into Sweden. And a synchrotron is essentially a huge microscope, or it can also act as a CT scanner. And what goes on is there are electrons that are running around this ring. This building is, I don't know, 200 meters wide, and it's running at the speed of light, or almost. And each time the electrons are bent, it sends out a very pure X-ray signal, which is then used at instruments around the ring. So we originally got money for this center from the capital region of Denmark, which is the political region around Copenhagen, to support research and industry's usage of this facility. Now the MAXIV was slightly delayed, so instead we practice in other ways. And I think one of the key things about infrastructure like this is to realize that this, and also the European spallation source, which is a neutron source that's built next to it, and similar sites around Europe and the world, is very useful for case studies. So you can take your specimen and put it into this huge microscope and get unique insights into the material. But what's really important, I think, is to somehow link it to the microscopes, etc., that are standing in the labs, in the basement of wherever you work. So this is the part that we've been practicing a lot, linking these large-scale facilities with the smaller facilities nearby. So that's the background. Now there's a whole bunch of partners, and I want to talk to you more about how we then, how we as image scientists have been supporting image researchers or biologists or material scientists on how to take images at synchrotron facilities and microscopes around the world, and particularly how to analyze the data afterwards. For one thing that is certain is that it takes a long time to produce the material that then is taken, that's imaged at various facilities, and very often these images are not put to full use simply because people underestimate the time and difficulties in the following analysis. So I've been working a lot with people from biology, with biological projects, and I think that's as a computer scientist, it's enormously inspiring because it's geometrically very challenging, it's computationally difficult, and it just allows me to, as a researcher, develop my field. But in this dialogue with, we call them users in lack of a better name, but image researchers, I've tried to list here what a typical focus have been from them, and that would be something like the cellular anatomy, or how things change over time, or even if you have a diseased or healthy population, can we come up with some way to analyze them to find a signal, that a biomarker that describes the differences. And what's certain is, what I see is, of course, that it takes a lot of work to make the materials to produce images, and often question is that they have is they have no clue from what's hard and what's easy in image processing. And actually, I also find that often the interaction I have with them is I ask really silly questions, and sometimes they can just tell me the answers, and other times, maybe I can offer a fresh perspective. On the other hand, as an image scientist, so someone who's processing images, as Stefan said, we do a lot of data cleaning and conversion. We do a lot of segmentation tracking and data analysis. And interestingly enough, maybe one of the key problems is for me to figure out what's biologically relevant, because I can think of many things that can be computed on images, but if it's not important, well, we shouldn't spend too much time on it. And zooming out a bit, the projects that we've found fruitful have been the ones where we are part in solving some problem, not just a computer science problem for the sake of computer science, but actually put computer science to use. And even more successful is when the methods we develop, that they can be used to in a broader context than just this one question. That's when I think I've succeeded. And as in part of that, I think it's really important that we train researchers, so train biologists, people that are working on material science, etc. What I think we should, when we do image processing, image science as a service, we should be very careful about choosing when to work on a graphical user interface. So I've been in the field of image processing for, I think, 25 years, and I've seen a lot of work been put into making nice graphical user interfaces. And the few that succeeds are, of course, set to use or well appreciated by the community, but lots of them don't. And I typically, when I try to make this perspective to my students, I show them my phone and just think about all the apps that have been developed which are not really used by anyone. And there's a huge risk that we spend our resources wrongly on this. I think also it's important that we as imaging scientists do not take over the analysis task of, for example, the biologist. The main reason being that, well, we might not know exactly what's relevant. And also, as I'll come back to, there's something about for whose sake we do it. The service that we've been able to offer has been for free. And I think for free is a fickle beast because a few times we've had the experience that users would like our help, but then when it didn't really turn out the way they wanted, they possibly thought they just gave us a toy we would play with. And this is a real risk that we have. Not that I don't like playing with methods and data. I like it even better if it's of use for someone or something. Now, the last point I have is this point about standard and simple tools, and I grade it a bit because I find that I often use standard tools and simple methods to open a dialogue with users. And it's back to this thing about what's easy and what's hard. Sometimes I decide to invest 3 hours in setting up a simple pipeline to test something really, really simple, and also to illustrate how far you can go if you invest sufficient resources. So the way we've done it, I think a structure similar to this is what Stefan was showing, and I actually think this is sort of being invented concurrently or at the same time many places around the world, also in all the collaborators at Euro-BioImaging. So I think this is really, it's a good time to discuss these things. But the way we did it in practice at the QIM service center was that we had 3, Stefan called it phases. So consultation, it's supposed to say there, with an expert. So that could be with me, for example. I sit and talk with people, they show me their data, or maybe they even show me their ideas for how images could be taken. And I often come with input such as, remember to use the full range of intensities, look at the histogram. I think this is an easy problem. I think this is a hard problem. In the QIM center, what we often use that for is a screening phase where we decide who has a project that is where it can be turned a fruitful collaboration and who should somehow be down prioritized. If the user is then elevated to the next phase, then we assign an image scientist. So that could be anywhere from a student programmer to a postdoc, or perhaps even me, that spends about 2 weeks of their time on working with the user and with their data. Not necessarily 2 weeks in time, but 2 weeks in total. If this sometimes turns into a publication, or it might turn into a decision on whether to extend the collaboration with an actual research collaboration, would typically mean that we would need to find funding. To support all of this, we hold workshops, PhD courses, and particularly we try to give the users the tool of Python. So the ability to do very simple stuff in Python, such that the methods that we develop, similar to what Stefan was showing you, is available to the users in Python. And it would typically mean that these 2 weeks service has resulted in a Jupyter notebook that is then either understood by the user or that they can work on the workshops. So when does all of this become a success? Well, some of the signs I've seen is that there must be a clear research goal. And it's, as scientists, it's a little bit funny that I have to stress this, but often, perhaps it's because we've been so much involved in the synchrotron community. Often there's a very happy-go-lucky feeling to the imaging being taken. Let's see what the synchrotron facility can show. But if there's no clear research goal, then there's no clear task either, and then the collaboration is not fruitful. There must also be a mutual drive. I can be as enthusiastic I want about doing image processing, but if it's not met by an equal enthusiasm by the biologist, it's not going to be successful collaboration. There must also be a match of skills, of course, it should be somehow a problem that's suitable for all involved. It should be somehow that, well, in my community, this is about furthering people's research careers. Somehow there should be a match. And it's also very useful to, at a very early stage, to talk about what's possible and what's not. So as my final point, I want to highlight some of the dilemmas that we in Denmark and also in Euro-BioImaging are talking a lot about these days. One of them is the finance model. So in the synchrotron community, this is led by physicists. Physicists are used to a finance model that, well, if you want to take an image at the MAX4, you make a research application. If this is then granted, then it includes the funding for taking the images. So you basically apply for money. In practice, you apply for money to do the images, but it looks like a research application. In biology, I think it's the same everywhere, but at least here in Denmark, people pay for microscope time. So they've, in their grant application, they factored in that they are planning on so and so many hours at a microscope at so and so cost. And this is one of the dilemmas, is how can we bridge these differences in traditions, but also how we can, let's say, convince people to pay for imaging science support when there's no hardware, it's people we're talking about. The second thing we're talking about is there are a lot of communities popping up in Europe and other places where people are sitting, as Stefan, I think, Stefan, what you showed was very impressive and actually quite a large effort, but there are a lot of resources available, but they're scattered around. I think it's really important that we gather forces, but also in a way that we avoid sitting in too many coordination meetings. So how is this done? This is something during my career I've seen people try many times in the software, trying to get together around a particular way of structuring software, and it's very difficult to do right. One could say that one of the reasons why Python is so popular these days is that there are all these libraries, but anyone who's tried to install anything in Python will realize that the plethora of developers doing their Python packages also means that it's very difficult actually to figure out to make all the pieces stick together. It's not a trivial task. And then the final thing I want to highlight is for people doing image processing as a service, there's always let's say, a balance between that. If it's a postdoc, then this postdoc has to think about his or her career, and when is service then to be thought of as teaching, or is it to be thought of as research? And I think that's a negotiation, as a separate negotiation in every project. So lots of things are happening. These are just some of the icons that are of things going around in the neighborhood of Copenhagen, and I'm sure you all have a similar wealth in opportunities. And I just want to go back to this one, saying I think it's really important that we collaborate. This is why I engaged myself in Euro-BioImaging. I think it's looking to become a really good platform. I'm looking very much forward to developing image processing and a service. It's an investment, but I'm sure it'll be worthwhile for all of us. Thank you.
Thank you so much, John. That was actually very inspirational, and I guess this is the kind of enthusiasm from image analysts and experts like you that keeps us going, actually. So it's very important for us. Thank you once again to both Jon and Stefan. And I am wondering at this point of time if there are any questions directly to the speakers. Sebastian, please go ahead, unmute yourself, ask a question.
Hi, thanks. Very interesting presentations. I'm just wondering, so these are very large projects that you've presented. It's not something that is just, let's say, run-of-the-mill analysis. What I'm wondering is, if, for example, I have someone from a small university who doesn't have access to a, let's say, imaging platform, or even better, data image analysis platform, and they want to do something that is clearly not a method that will be publishable. I mean, as you said, if it's, I can't give this to a postdoc because he doesn't have the time to do it. Is this something that would be part of the tasks of Euro-BioImaging, or is this not?
I don't know, maybe I can start to answer this and then pass it on to Stefan and Jon, because so the thing is that at Euro-BioImaging, we have facilities and who have image analysis experts, but also these people have different interests. So you heard from Jan today, he is really into developing software and very computationally intensive and interesting for that matter, analysis pipelines and tools. But we also have other facilities where we have some engineers who are happy to develop let's say if you want to segment your nuclei, I mean, sure, if maybe your data is too large and you're unable to handle them in a meaningful fashion, we would be able to find you a suitable facility and a suitable expert that can help you with your analysis. So it depends. So that's the kind of advantage we have with the richness of the people that are involved in this, because you will have each side of the spectrum. You will have dedicated Atlas projects where they're mapping every little synapse in your brain versus, hey, can you register 2 of these images for me? Okay, maybe that was a bit much. But I don't know, Jon, Stefan, would you like to comment on this?
Yeah, briefly. I mean, so at the population image node, I mean, we are open to small and large projects. So in that sense, we don't make a distinction. And that will generally be discussed in this take-in meeting, whether it's whether we can do anything. And I think, as Jon pointed out, it is of course nice if there is a prospect for publication, at least, if because then it could become interesting for a researcher, and then then it's not then it could become a research collaboration instead of like a really that someone really pays for all the hours. But if that's if there's no prospect for publication, then then it becomes more like a service that is being paid. But I would if you're saying that there's a small chance that at that center it becomes a publication, you could wonder, okay, what is the end goal? If it's not a publication, is it just for entertainment, or what's the goal?
Then I'll just maybe also start the video, because it feels a bit odd that I can see you and not the opposite. Hi, sorry. No, what I meant is that the method itself is not going to be something worthy of a publication.
Okay.
It is tricky. It involves adaptations using different filters maybe, but there's nothing really new in there. All the tools are around, and it's something that might take me, I don't know, a week or 2 maybe to make, and it's not going to revolutionize the field or whatever. It's just that it's not completely trivial, but it's also nothing extraordinary. So as you said, it's really the kind of service that a platform is providing. And considering how much the platforms, for what I know of them, are already pretty busy from the local work they have, I'm wondering if they have the time to actually take projects from the outside also.
Yeah.
So I'm not sure what I heard you say, but are you talking about that you are developing software that will be used by someone, or are you the one coming with the images?
So in my case, I am an image analyst on the platform.
Yes. So my view is that the image analysis as a service is similar to teaching. And at least my job, half of the time I'm asked to teach, that doesn't turn into publications very rarely. So I've already accepted that not everything is about publication. Mm-hmm. And we have been working with the management at our university on setting up deals that are, if we do service, we do less teaching. It's not everybody who can reach these agreements, but I think somehow that's one model we could use. There's also the, let's say, the payment model, which is one of the ones we are discussing. And so if somebody from external pays for my time, then again, that frees me from all the duties that otherwise follows with my job.
Yeah, but if I remember well, sorry, I'm getting you here for just a second. If I remember well, the cost of an engineer is going to be something like maybe 50 euros per hour, which is not something that the labs will be willing to pay if we tell them I have to work for a week on this. I'm not saying it should not. I'm just saying that whenever I was saying this to a researcher, they were like, yeah,
And
but I could also.
It's a really good point. And I think it's something that we need to discuss in more depth, because nothing is for free. And one of the problems I find is this, that in some communities, it's okay to pay a lot of money to access, top of the notch hardware. But then when we talk about top of the notch people, for some reason that's supposed to be free. And I think this is because in the research community, we happily help each other. And when is it not a friendly service, and when is it actually something that should cost something? And that's what we're discussing. And it becomes even more complicated if some people are from the industry and others are from the university. Then at least our university requires two quite different prices. And as a researcher, I just find that, oh, please, can't someone else figure this out? But I think, unfortunately, we have to engage in this discussion. Now, the final thing I want to say, Sebastian, is I actually often engage in these, on the surface, seemingly trivial tasks, because often I find that I get challenged in my abilities to do image processing, and I find gold nuggets that I have to develop. So even though it might seem trivial, I actually enjoy it, and to a large extent, I enjoy helping people.
I entirely agree, and I love my job, which is literally just this. It's just that I'm paid to do this, so it's fine.
Yes.
But if the university is paying me as a postdoc, they might not be fine with this.
They would also pay you for teaching. And one of the ways we try to sort of bridge this gap is that people, when we have hackathons or we have PhD courses or other kinds of teaching style events, is people, they bring their problems, and then we sit together when we try to empower them to sort of, do some of the stuff themselves, but also discuss, very code near opportunities in their problems.
Okay. So just to go back to the original question, if I know someone who's looking for support, let's say in Paris, where I'm not, they could contact one of the people from the nodes or that are in Paris, or how does it work?
Well, so I will first answer the question, and then I have a comment to what you were mentioning earlier. So indeed, so as of now, the image data services are not online, but we expect to have them by the end of this month, I would say, because it's just something that has to be incorporated. So there is a whole procedure for technology to be allowed to be offered through Euro-BioImaging, and we have completed the first step, which was showcasing, and now it has been approved to be available on the web portal as a proof of concept study for a year. So that should start this month, I would say. And then you should be able to simply go to our portal, and there would be a section on data where you can just access the data technology and ask for image analysis as a service. So if you know exactly which node you want to go to, you would be able to just go down the tree and select exactly the node you want. However, in another case, if you don't know which node you want to go to, you can just say, I have an image analysis problem. You would be asked to describe it a little bit, of course. And then the experts at the hub, together with the people at the nodes, would then decide who could be able to help you. And then you start a bit of a dialogue there in terms of analysis.
Okay.
Yeah.
Do you think maybe it's something that could be linked to ImageSC also, or it would be completely separated?
I mean, in a way, I think sooner or later, I mean, to be very frank, sooner or later, we would probably need something like a bit of a forum, whether it should be linked to ImageSC or not. I mean, why not? I find that like very useful. And I think half the bioimage analysts are probably already using it. So
at least.
at least, if not all. So yeah, maybe that, or another forum of some other kind, but we would definitely need to use something like that.
Okay. Thanks.
And one of the comments that I wanted to come back to was you did mention that that image analysts at universities are always so busy that can you really expect them to work on an external project? So, I mean, again, it would depend. It is very variable, because in certain cases you are actually allowed, for example, to take, let's say, 50% of your position funded by external projects, for example. So you can expect to then spend 50% of your time, just making this up, right? 50% is a random number here. So let's say half of your time taking up external projects. And I think what we want to really do by actually letting people take external projects is, often I have seen that people have this problem of, oh, it's an internal service. So people try to, they tend to not take it very seriously, or just be like, it's a friendly chat, and then it gets longer and longer, and hey, can you write more stuff for me? Indeed, within external projects, things tend to, or at least we expect that things will be a bit more defined. And then of course it will hopefully give something to the bioimage analyst to leverage their position in a way, because by the end you can show that, hey, I can work on these, I don't know, 10 external projects. 5 of them were just like, I don't know, a couple of hours each, and then 5 of them took me half a year or so. So this is the kind of something that we also want to promote, so that the bioimage analysts are also recognized a bit better in terms of the effort that they put into completion of a project.
Okay, thanks.
Yeah. And just as a side note, in France, you have a very competent French bioimaging association. They have this very cool acronym, FBI. I love it.
Lucky them. Yeah. Okay. Well, thanks a lot. So it's a lot of things that are in the objectives, but maybe not completely ready then for the, let's say, simple, and I'll put this in quotes, not interesting, but simple projects, because it's indeed fundings that have to be found, or adapting maybe contracts or things like this, also to free up some time for external projects.
I'm not sure I'm gonna agree. I mean, I wouldn't say that. In a way, if you come like to me on the 1st of June, let's say.
Okay.
No, kidding. My web portal team would not be very happy with this. But let's say we talk again in June, and you have some image analysis problem, and we open it up. I mean, if you already know where you're gonna go, for example, if you want to go to Paris, if you want to go to Jean-Yves' team, or I don't know where, then of course your project comes in. We send it to them and ask them like, hey, do you guys have the capacity to take it up right now? And if the answer is really yes, your simple problem can actually be solved fairly quickly.
Okay.
And just to encourage you a little bit more, there are initiatives that are being tested by different nodes. So also France Bioimaging, they tried this sort of an open house sort of a thing, where they invited some microscopists as well as image analysts, and they were helping people solve their image analysis problem kind of on the spot. So it's almost like trying to get things started at the moment. And I know that there is also some initiative by the BioIF. So in Sweden, Bioimage Informatics facility at Uppsala, they also run what's called the Call for Help, where they're able to help people on these small problems, just probably in real time, or who knows where things in image analysis take you.
Okay. Then thanks a lot.
You're most welcome. That was an interesting discussion. It does come up a lot, actually. We meet in an expert group, and often we end up discussing things along these lines. But it's always fun to hear all the different perspectives. Do we have any more questions or discussion points? Maybe I will ask a quick one to Jon, actually, because what? You're like, what? I actually wanted to understand from your point of view, do you think that there's something different inherently from the data type perspective between the images that are coming out of a microscope versus a synchrotron?
No.
No, perfect.
But I just see pixels.
I see pixels. I think we should write it down, or we write it on a t-shirt or something. That's fantastic. I see pixels. Oh, Ken, you have a question or a comment? Go ahead.
I was just wondering about whether how do you say it? You have analysis surfaces, and how does those images, when you finish or the analysis finish, connected back into the raw image, the raw data? How do you see the workflow, especially when you're talking about Euro-BioImaging and global bioimaging, that you're coming from, the services can be coming from different centers or different expertise. How do you see those things work together?
Any takers? Otherwise, I can start if you want. That is also an excellent question, Ken, because that also came up as we were having some of our internal discussions, that so far we've thought of a project. I mean, actually, often you would, even when you want the access to an instrument, you would apply to, let's say, access to one particular instrument. But let's say your project involves two different instruments. Well, I don't know, you can always go to two different instruments, right? And maybe do a first phase on one instrument and then on the second. So in a similar way, one could almost think of image analysis like that too. And since it tends to be a bit more modular, maybe this practice will become more and more common over time. So having, let's say, collaborative projects where maybe different analysts are working on the same problem, and maybe different parts of the same problem, to really make sure that everything fits together in a pipeline. So we've been toying with this idea. We'll just have to see if some projects come in that that actually require these kind of working together. I hope I answered your question, or maybe did I get it right, Ken?
Yeah, sort of. I think I think it's quite far-fetched at the moment, but is how do you the other thing is kind of like related to QualRAP, is how do you make sure those metadata and all those things are kept together, and how do you refer back to the original images and things like that. That is never very easy, especially, I mean, even within an institute, it's not easy with different groups, let alone external collaborations. I think it is management-wise is quite not so easy.
Maybe I can say a few words about that from our experience in the medical imaging domain. So this is exactly indeed what we are using XNAT for and Faster to manage this this data storage and seeing which results are derived from which image. So in XNAT, it can we start with the raw image data from the scanner, and then we add also the derived data to it in the same system, so you can really see, okay, this is a segmentation of this image, or this is a converted file from that DICOM file. So we keep the things together, and that really helps us to find back the data. And then in the Faster software that I presented, there's this automatic provenance that tracks, okay, where did you get the image from? And because the software directly loads it from XNAT, so there's no intermediate temporary storage. We can keep track better of the data flow.
And do you?
You're right, it's a challenge always.
Does XNAT also provide you access control or a lock of who access those data? Because I think you are dealing with human tissues and human images, presumably that you need to have more tracking of those things, or not? Yeah,
although we typically in research we anonymize the data as soon as possible, to not to make the restrictions less severe.
Okay.
But there's indeed also in XNAT some logging of access, but I don't think it's so detailed that you can really say, okay, that person downloaded that image at that day. It's not so exhaustive, but at least changes, they are logged to the images, so that already helps.
Okay, I should look at the XNAT thing then. Okay, thank you.
Yeah. It is a real problem about protecting privacy rights, and particularly here in Europe, we have the GDPR, which is causing us a lot of problems. As a researcher, but as a person, I'm very happy about it. I know my data is protected. And I think we are slowly in Europe figuring out how to do it well. but it feels like it's a problem that not quite been solved yet satisfactorily. I think though many of at least the material that I've been working on are far from people. So it's more of a research kind of material that makes it much easier. And I would also say if we were to engage in a let's say close to the hospital pipeline, maybe this community is a good place to start, but maybe it's not the intent of this data science as service. I think we should possibly talk in more detail about how to do it right on a large scale.
Yes, I mean, from a Euro-BioImaging point of view, there's also experienced medical nodes like where Stefan is associated, but also others who are dealing a bit more directly with these problems. And certainly XNAT is something that is helpful for them in general, but of course they have to do the data anonymizations and take care of those things for human images. And one more thing to add there would be that, I mean, currently we also at the Hub, especially in relation to the European Open Science Cloud projects that we are involved in, we are trying to make sure that we are developing further either the OME ZAR format or these kind of things where we want to be able to store this additional information within the file format for the annotations, inclusion of the annotations, as well as the metadata should be better, or at least that's the promise these NGFFs hold. So we are also exploring this very actively and working with the community to develop things further. So yeah, fingers crossed for that. Hope that works. All right. Do we have any more comments or questions at this point in time? If that does not seem to be the case, well, thank you all for joining us today and also for the lively discussion. And that was our intent. And it was very nice that we could show to you all the things that we're taking care of at Euro-BioImaging, all the fantastic services that our nodes are offering, and a very heartfelt thank you to both Stefan and Jon for being here today and sharing with us the great services and even the analysis procedures that they're following. So thank you for that. And if there's no more last comments, then.
Thank you for coordinating it.
My pleasure, really. All right. Well, get in touch with us if you want to hear more, and Euro-BioImaging.eu or info@Euro-BioImaging.eu, or even like personal email addresses, no problem. And we're happy to help you all with your image analysis problems. Take care and enjoy the rest of the meeting. Bye-bye.